So if you can't make my office hours today and you want to talk to me, please do send me an email as soon as possible so we can schedule a time. Thinking about the next exam. It is a week from Monday, April 18th. It's going to be on pathways. Start learning your pathways now. We're going to start glycolysis today. So it's going to cover the genetic membrane transport, proteins, and then as far as pathways as we get through. We certainly, I would imagine, glycolysis, fermentation, and the citric acid cycle. For the exam, so by next week I will have a study guide for you. I will have some practice problems up on Mastering Biology. For the review sessions, because the exam is on Monday, which means the exam material is on everything through lecture on Friday, which means you have a weekend, which is good to study for it. But for the review sessions, next week your recitation sections will count as review sessions. Right, so there won't be a quiz next week in recitation. It'll just be for you to go and review material for the exam. Okay, so that way everyone should be able to make it to a review session for any questions you have covering any of the pathways. Okay? Are there any questions on exam 3, exam 4, anything else related to the class before we get started? Quiet class today. Everyone's anticipating glycolysis, aren't you? All right, before we get into hardcore glycolysis and start looking at that pathway, I want to talk just a little bit about structure and function, because we're going to be talking about a lot of molecular conversions. We're going to be stepping through in all these pathways from one molecule to another. And in some cases, we're going to just be looking at, we go from this molecule to this molecule. But at each step, there's going to be an enzyme that is responsible for that change in molecular structure. So even though we are not going to explicitly talk about each enzyme, talk about a few as examples, some of the important ones, but at each step, that change in structure is controlled by an enzyme. So I just want to talk briefly about enzymes and what they're doing. So how does structure relate to function? Structure dictates function. The molecular level structure controls function. So the structure of your molecule is going to control what it is doing. We've seen lots of examples where we've seen examples throughout the class anyway, where single changes in amino acids of a protein can drastically alter its structure. Think about when we talked about sickle cell anemia. That single amino acid change went from a functional hemoglobin to, in the right environment, related to oxygen, a very significant change in structure of that protein, so it was no longer able to function. All right, so structure determines function. Why? Here's the bigger question. What about structure is determining that function? What's important if we think about proteins? Right, we're going to talk a lot about enzymes. We're going to look a lot about molecular changes in structure that are controlled by enzymes. So what about proteins is controlling function? What about that structure? What's the difference in amino acids? They have the same backbone, but different R groups. So those side chains, right? Those are the things that are controlling how that protein is interacting with other molecules. So structure determines function by positioning those functional groups. And the position of those functional groups and how that can interact with other molecules is determining the chemistry that can occur. We're going to look at some examples of that today. All right, so how do proteins, and particularly those side chains, those R groups, interact with substrates? What kind of interactions do you think? Hearing some whispering. What kind of interactions can molecules face? Hydrogen bonds, covalent or non-covalent? Non-covalent. What is another example of a non-covalent bond? A little bit louder. Ionic, dipole, so van der Waals, right? So lots of non-covalent interactions with those side chains. You guys are quiet today. Ionic bonds, hydrogen bonds, hydrophobic forces. Side chains can interact with molecules in all of these ways to control the chemistry that's happening. Of course, it can also form covalent interactions. So the side chains can interact in any possible way with the molecule to alter its function. That's what we're going to be looking at today. So how do enzymes specifically lower activation energy? They make the chemistry easy. They position the functional groups to interact with a particular molecule to move electrons around. All right, so they interact with something and set it up for a particular structural change. And in a lot of ways, we're going to see examples today of moving electrons. So we're going to be talking about producing energy for the cell. And that is driven by moving electrons to different molecules and moving them across the membrane. We're tracking what happens to our energy molecules and where electrons are moving. Okay, so I just wanted to give you a brief introduction since we're talking about a lot of moving from one molecule to another. It's always controlled by an enzyme. We're only going to talk about a few of those enzymes in detail, but at every step there's an enzyme controlling that structural change. All right, so that leads us to glycolysis. We're going to start talking about energy metabolism. We look at a cell. We have a cell here. This represents a mitochondrion. The cell picks up things from outside and moves it in. Things can be degraded back to their functional units. So amino acids play a role in multiple steps in this cycle. Fatty acids and glycerol, we're going to see in a number of places. But where we're going to start is with simple sugars. Glucose. So we talked about membrane transport and how glucose is moved into the cell. Now we're going to talk about what cells do once it gets that glucose inside. How is it using that to produce energy? We're going to go all the way from glucose through pyruvate, through acetyl-CoA, to the citric acid cycle, to electron transport, where we are producing our ATP. We're going to look at each one of those phases, each one of those steps in detail. We're just going to focus on once glucose gets into the cell, what happens to it. So if we think about cellular respiration, we're talking about mitochondria here, so we're talking about eukaryotes. Cellular respiration happens in the presence of oxygen. We're going to look at pathways after this that can produce energy in the absence of oxygen. But everything we're going to look at for the next few lectures, cellular respiration happens in the presence of oxygen. And there are 4 linked pathways that we're going to talk about that you're going to be responsible for knowing. Those 4 steps are glycolysis, where we take glucose to pyruvate. Pyruvate processing, where we take pyruvate to acetyl-CoA. We're going to talk about the citric acid cycle, which starts with that acetyl-CoA and produces some electron carriers plus carbon dioxide. And then finally, that electron transport chain and chemiosmosis, where we're producing most of the ATP out of this whole entire process. So at each step of each pathway, I'll tell you what you need to know, but you're going to need to know the steps in that pathway. In some cases, you're going to need to know the structures. And in every case, you're going to need to track where ATP is used, where ATP is produced, and how many of those, and track your electron carriers. So if we look at these 4 steps, they happen in different cellular compartments. Glycolysis, this first step that we're going to look at today, happens in the cytosol. So a cell brings glucose in, and it gets processed right away in the cytosol to pyruvate. And then that pyruvate is moved into the mitochondrion, where the remaining steps in cellular respiration occur. If you want to look at a video, there are lots of them on YouTube. Here's one example that will be in the lecture. I'll post on Facebook. This goes over not necessarily the detailed steps, but what happens in each one of those linked pathways and where our energy molecules are produced. So this is an overview of that. And we're going to look at each one of these in detail over the next few lectures. All right, so we're going to start with glycolysis, taking glucose to pyruvate. All right, so you should be able, once we get through glycolysis, to fill in this. All right, this is showing all of the arrows for all of the steps in glycolysis. So once we're done going through this, you should be able to draw this out. And I'll show you the filled-in version of it in just a minute, but we're going to take it piece by piece.
Yeah, right? So what?
You should also be able to draw structures from glycolysis. We're going to go over the molecules.
So like just the
These are some of the examples of the molecules you should be able to draw. And if you understand the pathway and what's happening, then you should be able to go from one molecule to the other. All right, we're going to start looking at that today. You don't need to know it now. This is what you should be able to do by the end of the section on glycolysis. Yeah, oh yeah. All right, so that is all the way from glucose to pyruvate. Notice that it splits the molecule in half, and then you end up with 2 pathways, or going through the pathway twice for each one of those molecules. So you have to keep track of that in your counting. Some of these molecular names are a little long, so I've used abbreviations, but you need to know the full name of those as we go through. We're going to divide this down into phases to make it a little bit easier to handle. Phase 1, phase 2, and phase 3. So phase 1, taking glucose to glyceraldehyde 3-phosphate. Taking that G3P down to 3-phosphoglycerate. And then all the way then from there to pyruvate in phase 3. Notice that in phase 1, I'll stress this again, we are using ATP. So that's the energy investment phase here. Whereas in phase 2 and 3, we are creating ATP as well as some electron carriers. Keep track of where ATP is invested and where ATP is produced. Okay. Don't worry about getting that all down. We're going to go through it step by step. And we'll look at the full thing again at the end. All right, so we're going to start with phase 1. Start with glucose. Step 2, glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate to G3P and D-hap. We're going to look at each of those steps in terms of the structure of the molecule and what the structural changes are in each step and where ATP comes in. So I want to start off with our glucose. Excuse me. Glucose is often drawn as a 6-carbon ring. We are going to look at it in the linear version for this pathway. Bless you. So the very first thing that happens in glycolysis, once we get a glucose molecule into the cell, in the cytosol, we use a molecule of ATP to get it going. So to go from glucose to glucose 6-phosphate, we hydrolyze ATP to ADP. What does glucose 6-phosphate mean? Where have we put a phosphate onto our glucose? The 6-carbon. Pretty straightforward. Where did that phosphate come from?
The monophosphate that came off.
It came from our ATP, so it's the phosphate we cleaved. When we went from ATP to ADP, we cleaved off a phosphate and stuck it onto our glucose. So step number 1, use an ATP, hydrolyze that 3rd phosphate, and use it to make glucose 6-phosphate. All right, step 2, we're going to go from glucose 6-phosphate to fructose 6-phosphate. What's the structural change you see between these two molecules? What's changed position? The carbonyl is flipped with our hydroxyl. All right, so we've gone from an aldehyde to a ketone. To go from glucose to fructose. So we just moved electrons around in this particular step. Okay, step 2. So step 2 is simply a structural rearrangement with the molecule we have. Nothing's been added. We've just moved things around. Go from glucose to fructose. All right, step 1, glucose 6-phosphate. Step 2, glucose 6-phosphate, fructose 6-phosphate, just a structural rearrangement. Step 3, fructose 6-phosphate, we're starting with as our substrate. We invest another ATP to go from fructose 6-phosphate to fructose 1,6-bisphosphate. So similar to step 1, we're stripping a phosphate off that ATP. We're using the energy and the phosphate to add that phosphate to the molecule that we're manipulating. Where does that phosphate go? Which carbon? First carbon. Fructose 1,6-bisphosphate. 2 phosphates, 1 on the 1 carbon, 1 on the 6 carbon. All right, so we've added a phosphate. We started with glucose, we added a phosphate, we rearranged the structure, we added another phosphate. So in glycolysis, from glucose to fructose 1,6-bisphosphate, We have used 2 ATP in this first phase. And we are transferring groups. Why are these necessary? Why do we want to stick phosphate on there? All right, ponder that as we look at what happens to it next. We'll come back to that. All right, think about why we need to add ATP to get this whole process going. All right, that's where we are. We're at fructose 1,6-bisphosphate. Next step. We're going to cleave that 6-carbon structure into 2 3-carbon structures. We're going to go from fructose 1,6-bisphosphate dihydroxy acetone phosphate, or DHAP, and G3P. DHAP, glyceraldehyde 3-phosphate. So how are those 2 molecules different now? What's the difference between G3P and DHAP?
They split it in half.
Split it in half, but structurally, how are they different? The placement of the carbonyl. We've already seen an example of how we can flip that around, right? So these are 2 very similar structures. We've looked first at DHAP, right? Just illustrate how it's named. So that's acetone. That's dihydroxy acetone. What we have at this step is dihydroxy acetone phosphate. Or DHAP. Second molecule is based in glycerol. We have a phosphate on there, so this is glyceraldehyde 3-phosphate. So those are the 2 molecules produced when we split fructose 1,6-bisphosphate. The specific enzyme, if we look more closely at how this happens, that's controlling cleaving our fructose 1,6-bisphosphate, our 6-carbon molecule into 2 3-carbon molecules, is fructose bisphosphate aldolase. So let's look a little bit more closely at how that enzyme is achieving that cleaving of the structure. So aldolases, they're enzymes that catalyze the cleavage of a 3-carbon glycerone unit using a Schiff base. So the first thing we have to figure out is what a Schiff base is. Anybody have any ideas? You should know. Right. It's amine. It involves a nitrogen. So it's a functional group that contains a carbon-nitrogen double bond. With that nitrogen connected to either an aryl or alkyl group, but involves a nitrogen. So if we know that it involves a nitrogen, what amino acid, right, means that our enzyme has to have a nitrogen somewhere in the binding site. It's going to interact with this molecule. Think about your amino acids and their side chains. We know that the aldolase is cleaving that structure using a shift base, which involves a nitrogen. Which one of those amino acids has to be involved?
Okay.
Think about your amino acid structure.
Oh yeah. Okay.
Think about the side chains on each one of those amino acids.
4. The K has 3 and then this is 1. And then it has that weird one? That has like, yeah, like a C and then an N. And then like that, yeah. And then like, wait.
One has a nitrogen. Can't forget your amino acid structures. Okay, everyone have a chance to answer? I'm going to run through their amino acid structures. Take a look. All right. Good. You remembered your amino acids. Everyone's awake today. Lysine is the only amino acid up there that has a functional group with a nitrogen in it. All right, so it has that functional active site in the enzyme. If it's going to form a shift-base linkage with our molecule, it has to involve a lysine of that set. All right, so we looked at how that happens. Our enzyme, our aldolase, has a lysine in the functional site. It's going to form a covalent intermediate, with our fructose 1,6-bisphosphate. So we have here our nitrogen, double bond, shift-base linkage. That's our lysine R group. And then the rest of our enzyme. Does everybody see that? All right, and through that temporary covalent intermediate, that shift-base linkage, the enzyme can hydrolyze the 6-carbon structure into 2 3-carbon structures. Okay, so we've gotten to DHAP and G3P. So I've already pointed out they are pretty similar structures, different in where a double bonded oxygen is in the structure. And so with a rearrangement of the electrons, DHAP can be flipped back and forth to G3P. So in our first phase of glycolysis, we've gone from glucose, added 2 phosphates to the input of 2-ATP, and then we've cleaved fructose 1,6-bisphosphate into G3P in DHAP. DHAP, with an rearrangement of electrons, can be interconverted to G3P, so that in the end, we produce 2 of the same 3-carbon molecule, G3P, glycerate 3-phosphate, glyceraldehyde 3-phosphate. So now the pathway, because we have 2 of the same molecule, everything that happens is going to happen twice, once for each molecule of G3P that we've produced. So one of the reasons that this first phase happens in the cytosol is because as soon as we bring glucose into the cell, we convert it to G3P, or to even just to G6P, glucose 6-phosphate. What does that do to glucose concentrations? Keeps them low. So we can continue to bring glucose in using our carrier-mediated diffusion transport protein. So if we bring glucose in, we add a phosphate to it, and start that process in the cytosol, as soon as it gets into the cell can control concentrations of glucose to continue bringing glucose in across that membrane. It also keeps that glucose from being shuttled back out due to diffusion processes. All right, so as soon as the cell gets a hold of the glucose, it's going to start this first phase of glycolysis, if it is in the presence of oxygen. Okay. So phase 1, glucose to 2 molecules of G3P. So that takes us to phase 2. Phase 1, energy investment phase. Phase 2, we start getting energy back out of our system, either in terms of ATP or in terms of NADH, which we're going to take a closer look at either today or first thing on Friday, which is an electron carrier. Remember, this whole process ends in the electron transport chain. Even if you don't know what that is yet, you know it involves electrons. So we're dumping electrons into molecules that we're going to send to that electron transport chain. Okay, so in phase 2, each process happens twice, once for each molecule of G3P. We're going to go from G3P to 1,3-bis phosphoglycerate. So what happens in this step? We're adding another phosphate. Where is that phosphate coming from in this instance?
ATP.
Are we investing any more ATP? We're starting to produce it now, right? So past that first phase, we are adding phosphates from inorganic phosphate. We're not stripping it off of ATP. So in phase 2, we're going from G3P to 1,3-bis phosphoglycerate. And in that conversion, we are adding an inorganic phosphate to our molecule. And we are rearranging our electrons in that process to dump some electrons onto our electron carrier to go from NAD+ to NADH. NAD stands for nicotinamide adenine dinucleotide. If there's an H on it, then it's nicotinamide adenine dinucleotide plus hydrogen. All right, nicotinamide dinucleotide.
We're still gonna.
Dinucleotide because we have 2 nucleotides covalently linked by their phosphate groups, so phosphate bridge. One of those nucleotides has adenine. The other one, the base, is a nicotinamide. The relevant parts of this that we are most interested in is that nicotinamide. This is the part that's carrying electrons around for us. So it comes in an oxidized form or a reduced form. So it can pick up electrons and move them around for us. So if we look specifically at the reaction that we're considering now, how is that NAD playing a role? All right, so let's take a closer look at that for the last few minutes of class. So we're going from G3P to 1,3-bisphosphoglycerate. Here's our glyceraldehyde 3-phosphate. Here's our enzyme that has an NADH or an NAD in the functional site. So GAPDH, glyceraldehyde phosphate dehydrogenase, which has that NAD as part of its functional site. What else do you see as part of that functional site? What amino acid residue? All right, think about your amino acid structures again. We've got an NAD. What other functional group is shown there? What amino acid has to be involved in this particular molecular structural rearrangement?
Why is it in this? Oh yeah.
Everyone have a chance to answer? Which amino acid structure has to be involved? Or amino acid side chain or R group? Let's take a look. All right. Everyone's got their amino acids down. It's cysteine. Cysteine is the only one up there that has that sulfur as part of its R group. All right, so let's look at how this enzymatic reaction occurs to move our electrons around and end up adding that inorganic phosphate. So we have our enzyme, NAD+, we have our G3P. We form a covalent intermediate between that cysteine side chain and our G3P. You know what's coming next, right? What kind of bond are we looking at? This one you guys may not be familiar with, so I won't ask you for this one. All right, take a look at it though. So this is called a hemiacetal linkage. It's derived from an aldehyde with the addition of an alcohol to that carbonyl group. So it looks something like that. So we formed a hemiacetal linkage, except ours actually looks like that. What do we name things when we have a sulfur involved? What's the functional group? A thiol. So this is actually a thiohemiacetal linkage. So the first step is we form this thiohemiacetal with our dehydrogenase, our enzyme. Once we have that intermediate, we move around some electrons. Right. We transfer our hydrogen to our NAD. form a carbonyl with our oxygen? All right, so that particular structure, now that we've rearranged our molecule, what kind of bond is that? A little bit different than what you're used to seeing for this bond type.
This is on Facebook. It's just a suggested post. Apparently, it must be
It is a bond type you know, though.
Oh, this is real? Or you didn't know the other Wow. So it's
What would you call it if, instead of that sulfur, you had an oxygen there?
I have no idea. This is Mandarin.
You know, like the
Alright, everyone gets their answer in. Look at the answer. Before everyone takes off today.
Yeah.
Alright, let's take a look. Alright, so it is an ester. So the answer is C, it is an ester. What do you call it? Is there sulfur there? Thioester. Alright, we'll pick up there on Friday to get to the rest of glycolysis. Start learning your pathway, start learning your molecules. Alright, each step, you break it down, it's not too bad. Adding a phosphate or rearranging the
to change it and print it out.
Have a good Wednesday.
Right here
Your
on
homework
media.
for 14.
Do you want to look over it to see? Oh my gosh. Okay, I'll look over it. Oh, yeah,
Yeah, it's okay.
that's